హాయ్ ఫోక్స్, వెల్కమ్ టు మై ఛానల్ ది యావరేజ్ డర్మాడాక్. ఇక మొదలెడదామా? Zn staining, Ziehl Neelsen staining, Ziehl Neelsen, Neelsen, Ziehl Neelsen staining, also known as acid fast stain. What is meant by acid fast? Ability to resist decolorization or ability to retain its red color, that is primary stain, carbol fuscin, when treated with acid or alcohol. Bacilli will retain its red color even with treating with acid or alcohol. It won't get decolorized. During decolorization, acid fastness is because of high content of lipids, mainly mycolic acid. In my previous video I told you mycolic acid is present in cell wall. Where inside cell wall? In electron transparent zone. Inside electron transparent zone, inner leaflet. I have drawn and shown you thread like structures in pink color. Both leprae and tuberculosis are acid fast as well as alcohol fast. However, M. leprae is less acid fast or alcohol fast than Mycobacterium tuberculosis. M. leprae can resist decolorization with acid of up to 5% H2SO4, whereas M. tuberculosis can resist up to 20% sulfuric acid. 20% sulfuric acid for Mycobacterium tuberculosis, 5% sulfuric acid for M. leprae. If we use more than 5% of sulfuric acid, it will get decolorized and everything will appear as blue because of counter stain methylene blue. What are all required to do Zn staining? Materials required. First, sample, nothing but slide with fixed smear. I already told you how to do slit skin smear. That smear. Slide stand to keep the slide. Bunsen burner, blotting paper, immersion oil, primary stain carbol fuscin, which gives red color to bacilli. Decolorizer, either 5% sulfuric acid or 1% hydrochloric acid in 70% absolute alcohol. Counter stain, 1% methylene blue, low efflux methylene blue. Method of staining: take the slide with fixed smear and place it on staining rods over a sink. Primary stain is nothing but first stain which we will use. Flood the slide with freshly prepared or filtered carbol fuscin. If it is not freshly prepared, already prepared one, then just filter it and use. Alternatively, gently heat by moving the Bunsen burner from one side to the other below the slide, just to produce steam to rise from all parts of the slide. Avoid boiling and keeping the flame static at any area under the slide. We have to keep on moving the Bunsen burner from one side to the other side below the slide until steam rises from all parts of the slide. Then we have to stop it. Avoid boiling and keeping the flame static at any area to avoid localization of the heat and boiling in that particular area. And then keep the stain for about 20 minutes without any further heating. And finally, wash the slide gently under running water. Primary staining finished with carbol fuscin. Then decolorization. We will try to de-stain the slide using, already I told you, 5% sulfuric acid. How much time? 10 minutes. If 5% sulfuric acid is used, we have to de-stain the slide by flooding the slide with 5% sulfuric acid for 10 minutes. If 1% HCl is used, 3 to 5 seconds based on thickness of the smear. If less thick, 3 seconds is enough. If the smear is more thick, 5 seconds we have to apply until the slide is faintly pink. Whenever you observe the slide is turning into faintly pink color, then immediately wash gently with running water again. Now primary staining finished, decolorization finished. During decoloration, mycolic acid has already bound to primary stain carbalfacin and became red. It won't leave that stain because it is acid fast and remain as red color only. Now we'll do counter stain with 1% methylene blue for 10 seconds. If we are using 0.2% less percentage of methylene blue, we have to keep for 1 minute and then wash under running water gently, then allow to dry, keeping the slide inclined on its narrow side on a blotting paper. Except emblepre, remaining all other structures would have decolorized using acid. Now it will take methylene blue and become blue. So we will observe lepra bacilli as red rod shaped structures against blue background because of staining with methylene blue. Now slide is ready for examination under light microscope under oil immersion objective. List of acid fast organisms. In Mycobacterium species, pathogenic Mycobacterium leprae, tuberculosis, Marinum, scroflatium. Non-pathogenic Fortunatum indicus prani, Mycobacterium indicus prani, which is used for preparing vaccines for leprosy. Next, head of sperm, bacterial endospores, oocysts of Isospora, Cyclospora, Cryptospora, Nocardia and Rhodococcus. All these are also acid fast. Remember the channel name, The Average Dermadog. Don't forget to like, share and subscribe. Thank you. Hasta la vista. Das vidania.